Welcome back to AllAboutRx. Today, we are breaking down a seriously vital metabolic pathway, the hexose monophosphate shunt, or as you probably know it, the pentose phosphate pathway. We're going to explore this whole sequence together right now, so you walk away with a crystal clear understanding of how your body manages energy and, really importantly, cellular defense. Okay, let's dive right into this. Our roadmap today goes from HMP Shunt Basics into the two core phases, down into the clinical relevance and pathology, and we'll wrap up with a high-yield quick recall. Starting off with Section 1, HMP Shunt Basics. So I want you to think about this for a second. What actually happens when your body needs more than just ATP? We know glycolysis and the citric acid cycle are basically the heavy lifters, right? They handle about 90% of your glucose oxidation. They're the powerhouses. But our cells are like these bustling microscopic cities. They don't just need electricity to run. They need raw building materials and heavy-duty defense systems. So the body absolutely has to have an alternative route for these really specific non-ATP needs. And that alternative route is the hexose monophosphate or HMP shunt. You'll definitely hear it called the Pentose Phosphate Pathway too, or even the 6-phosphogluconate pathway. Simply put, it's the premier alternate route for glucose utilization. And, location is everything in biochemistry. This entire pathway happens exclusively in the cytosol of the cell. It totally bypasses the mitochondria. Now, because of what this pathway actually makes, it isn't equally active everywhere. You'll find exceptionally high HMP shunt activity in places like the liver, the lactating breast, adipose tissue, the testes, and the adrenal cortex. So, what do all those have in common? Well, they're all sites of massive lipid or steroid hormone synthesis. They are building really complex molecules. And the HMP shunt provides the exact raw materials they need to get that construction done. Okay, quick pause for a massive warning here. If you're taking an exam anytime soon, you need to literally etch this into your brain. Absolutely no ATP is consumed or produced directly in this pathway. None. Zero. It's a classic trick question to ask how much energy, as ATP, gets generated here. The answer is always zero. This pathway is completely focused on making other vital molecules, not standard energy. Moving on to Section 2, the two core phases. It really helps to mentally color code these two halves so you can keep the sequence straight. Phase 1 is the irreversible oxidative phase, meaning once glucose enters this phase, it's fully committed. There's no going back. Phase 2, on the flip side, is the reversible non-oxidative phase. This back half is totally flexible, which lets the cell adapt on the fly to whatever it needs in real time. Now, what's really fascinating here is the very first step. The enzyme glucose 6-phosphate dehydrogenase, or G6PD, is the ultimate gatekeeper. It is literally the rate-limiting and regulatory step of the entire pathway. It grabs our starting material, glucose 6-phosphate, and oxidizes it. Let's break down this irreversible first phase step by step. First, G6PD oxidizes glucose 6-phosphate. Crucially, while doing that, it reduces a molecule of NADP+ into our very first NADPH. Next, an enzyme called lactinase steps up and hydrolyzes that product to make 6-phosphogluconate. And finally, step 3. 6-phosphogluconate dehydrogenase comes in for an oxidative decarboxylation. That's a mouthful, but it basically means we lose a carbon as CO2, generate our final phase 1 product, ribulose 5-phosphate, and boom, we get a second molecule of NADPH. So out of phase 1, we net 1 ribulose 5-phosphate and 2 highly valuable NADP molecules. All right, let's look at how this feeds into the next part of the story. This is phase 2, the broader reversible non-oxidative phase. Where phase 1 was all about pumping out NADPH, phase 2 is super active in cells that are busy synthesizing nucleotides and nucleic acids. You can kind of think of phase 2 like a magical biochemical recycling center. It uses key enzymes with names like epimerase, ketoisomerase, transketolase, and transaldolase. These guys are basically molecular jugglers. They seamlessly interconvert sugars ranging from 3 to 7 carbons. So if a cell needs to divide, it takes the ribulose 5-phosphate from phase 1 and morphs it into ribose 5-phosphate, the absolute essential building block for DNA and RNA. But because this is entirely reversible, if the cell already has enough nucleotides, no problem. It just juggles those carbons around and drops them right back into normal glycolysis as fructose 6-phosphate and glyceraldehyde 3-phosphate. It's just a beautifully efficient system. Section 3, Clinical Relevance and Pathology.
So the crucial point here is understanding exactly why your body is so desperate for these specific products. Let's weigh them against each other. On one side, ribose 5-phosphate. Without it, you are not building nucleotides, nucleosides, nucleic acids, ATP, or coenzyme A. Cell division would literally grind to a complete halt. On the flip side, we have NADPH. This is a massive reducing agent. You absolutely need it for synthesizing fatty acids, cholesterol, and steroid hormones, which, remember, is exactly why we saw high HMP shunt activity in the liver and adrenal cortex earlier. But here's the real kicker. NADPH is what maintains a molecule called reduced glutathione inside your red blood cells.
And that brings us to a really severe clinical condition, G6PD deficiency. You remember that rate-limiting enzyme from phase 1, right? Well, if a patient has a genetic deficiency in G6PD, this whole pathway creates a massive bottleneck. The immediate domino effect is a huge drop in NADPH production. And without enough NADPH, those red blood cells simply cannot maintain their reduced glutathione. Without glutathione, the cells lose their primary antioxidant defense system. They are left completely defenseless against oxidative stress, which leads to their rapid destruction. We call this hemolytic anemia. The crazy part is, a patient with G6PD deficiency might feel totally fine day to day, until they hit a precipitating trigger that just overwhelms their weak antioxidant defenses. You definitely want to know this trigger list. First up, oxidant drugs. We're talking antimalarials like primaquine, NSAIDs like high-dose aspirin, and antibiotics like sulfamethoxazole or chloramphenicol. Second, infections. Your body's own inflammatory response actually generates free radicals that can damage those vulnerable red blood cells. And third, this one always surprises people, certain foods, specifically fava beans. Eating them can trigger a full-blown hemolytic crisis, a reaction historically known as favism. Section 4. High-yield quick recall. Let's mentally build your ultimate flashcard right now to lock in everything we just went through. Let's summarize. The pentose phosphate pathway happens entirely in the cytosol. The golden rule, no ATP is used or produced. The big rate-limiting gatekeeper is G6PD. The oxidative phase is strictly irreversible and hands us 2-NADPH and the ribulose 5-phosphate. The non-oxidative phase is fully reversible, using transketolase and transaldolase to juggle things around. End of the day, we get ribose 5-phosphate for DNA and NADPH for fat synthesis and cellular defense. And if that G6PD is deficient, watch out for hemolytic anemia triggered by oxidant drugs, infections, or, fava beans. I want to leave you with a thought to really tie this all together. How wild is it that simply asking about a patient's diet, like finding out they love fava beans, could literally save their life? By understanding this invisible microscopic world of the HMP shunt, you get the power to predict and actually prevent a massive macroscopic event like a hemolytic crisis. Biochemistry isn't just a bunch of textbook pathways to memorize, it's literally the baseline language of human health. I hope this explainer sparked your curiosity and made things crystal clear. Keep studying hard, and we'll see you in the next explainer.